Chapter 1: The Scientific Study of Life. In this course, biology is defined as the scientific study of life. This includes all forms of life, going from little tiny bacteria to organisms that you're more familiar with, such as plants and humans. Biology also includes the interactions that occur between all the various forms of life, not just a study of one organism. You'll see throughout this course that the field of biology is constantly changing as new things are being discovered. For example, when we talk about the categorization of living organisms, there are new categories that have come up only in the last few decades due to new advances in DNA technology. You'll discover that the science of biology is constantly changing. To start off, we ask the question, what do all living organisms have in common? One important feature of life is that all organisms are made of cells. Think of any organism that comes to your mind. By definition, that organism would be composed of either one or more cells. In future weeks, we'll talk about the components that are found inside those cells. When you look inside each cell, you also discover that every organism has something called DNA. This DNA is passed on from one generation to the next and provides the information needed to produce proteins. We'll be talking more extensively about what a protein is, how it is made, and what the function of a protein is in living cells. Look at this picture of an ecosystem. You see both the living and non-living components in an area. What are some of the things that are alive in this picture? What about things that are not alive? How do you know that the lions are alive? What about the rocks? Are they alive? Why not? At first glance, you might think that it's easy to determine whether an organism is living or not living. But in some cases, this is actually harder than you think. Think about how you determine if something is living or not. Some things are easy to determine, but others can be a little tricky. What about something like a virus? Is a virus alive or not? Some may say that it's because it has DNA or RNA and can reproduce itself in other living organisms. However, it doesn't have one of the main characteristics of life. Viruses are not composed of cells. In fact, many viruses are just made of proteins and DNA. So it becomes important for scientists to come up with a definition or characteristics of what constitutes life. Scientists in biology have come up with 5 shared characteristics that all living organisms must have. The first is that life is organized in a very specific way. Life is highly organized and has a very specific hierarchy that starts with atoms, the smallest chemical unit of a living organism. In fact, all matter, living or non-living, are composed of atoms. It's the most basic unit of structure for all matter. Next on the life organization hierarchy is a molecule. Molecules come about when atoms are joined together in various ways. In later chapters, we'll learn about the different bonds that occur between atoms that give rise to molecules. When certain molecules come together in very specific ways, it can become an organelle, a membrane-bound structure that has a specific function within a living cell. Not all cells have organelles, but eukaryotic cells, the ones that have their DNA in a nucleus, have organelles in them. One example shown here is an organelle in a plant cell called a chloroplast. Other organelles include mitochondria, endoplasmic reticulum, ER, Golgi apparatus, and so on. At the next organization of life, we encounter the fundamental unit of life, the cell. This cell can include organelles and specific molecules that are housed within a plasmid membrane. Again, we'll be learning about various organelles in a cell, as well as the composition and function of the plasmid membrane. Now, lots of cells can also come together to perform a certain task in a living organism. When the same type of cells work together to complete a specific function, we call these groups of cells tissues, a collection of specialized cells. This picture shows the epidermis of a leaf as a tissue because it has a collection of cells that function together in photosynthesis. Then, when you have two or more tissue types that interact and function together, you form an organ. In this example, a leaf would be considered an organ because it's made up of cells that come together as tissues to form a leaf. You're probably more familiar with organs like a heart or lungs. By definition, a leaf actually is an organ because it's composed of two or more tissues. Now, many organs can then come together to form an organ system. In this case, the above-ground parts of a plant work together to absorb energy from the sun and transfer this energy to different parts of the system. Again, you're probably more familiar with organ systems like the digestive system, where several organs come together to form the digestive system, organs such as the stomach, small and large intestines, the colon, and so forth. Then we finally get to the organism, a single living individual. All the organ systems collectively make up this organism. In this case, it's an acacia tree. An individual organism comes in various forms. There are ones that are single-celled, like bacteria and amoeba, and then there are organisms that are multicellular, or composed of many cells. You're probably more familiar with these types of organisms, ones like animals, plants, and mushrooms. There are even organisms that are composed of mainly tissues, ones like jellyfish and worms. More complex organisms like you and me have organs, tissues, and organ systems. As I mentioned earlier, biology is a study of living organisms, but this also includes the interactions between organisms. So in the next level, we're at population. This is a group of the same species of an organism that live in the same area. So that means there are a whole bunch of organisms, but they're all of the same species. When we talk about evolution by natural selection, we'll be referring to populations, groups that are all composed of the same species of organisms. Now, when you have many populations of living organisms occupying the same region, we call this a community. In a community, you have two or more populations living in the same geographical area, such as all the populations living in a savanna. Then we get to the ecosystem, which includes both the living and the non-living components in that geographical area. That would include the rocks, waters, mountains, and other non-living parts of the system. Lastly, the biosphere is the most inclusive category, encompassing the entire global ecosystem. This includes the atmosphere and the entire planet where life is possible. Now, one interesting aspect of this organization of life is that living organisms have the capacity to show emergent properties at every level. What does this mean? It means that the components interact in a way where the original characteristics of each feature changes when it moves up to the next level. Here's an example of a brain cell. By itself, it serves certain functions as a cell. However, when many brain cells come together to form tissues, and tissues form the organ, brain, properties such as consciousness and memory emerge. The brain cell itself did not have these properties, but working together in complex networks, these brain cells that make up the brain have new emergent properties. Now we're ready to spend a couple of minutes to see if you understood the concepts that we've just discussed. Go ahead and pause the video and answer the following questions. We have now completed the first characteristics of life, that life is organized. So then the second characteristics of life is that life requires energy. As you see in this figure, the main energy source comes from sunlight. We categorize living organisms based on how they obtain their energy source. Producers extract energy from sunlight and make their own source of energy in carbohydrates, specifically glucose. Consumers are living organisms that get energy and nutrients from eating other organisms. Then lastly, decomposers get nutrients by externally absorbing their food from dead organisms and organic wastes. Notice that heat energy is lost every time energy is transferred from one organism to another. So all living organisms fall into one of these categories when it comes to energy. All life forms obtain nutrients from their energy source and use energy in order to live. The third characteristics of life is the necessity to maintain internal constancy. We call this homeostasis, which is a process where cells or organisms stay in equilibrium. A thermostat is a good analogy of homeostasis in action. Let's say that the room is getting too warm. If that happens, there's a sensor in the thermostat that opens the switch and turns off the heater, and then the room goes back to equilibrium. But then let's say that the room starts getting a little too cold. Then the thermostat readjusts and the switch closes and the heater turns back on. The room maintains homeostasis by staying at a specific range of temperature. Humans have an internal thermostat that helps us maintain homeostasis. Does anyone know what our body temperature is? Right, it's around 98.6 degrees Fahrenheit or 37 degrees Celsius. What happens if our body temperature goes up 5 to 8 degrees? You probably know that we can't go too far away from our body temperature, especially for a prolonged period of time. If we were at 103 degrees for more than a few days, this could cause damage in many of our cells and potentially kill us. So humans have various ways that allow us to maintain our internal body temperature. When we get cold, we start shivering so that more heat is released from the motion of the skeletal muscles. Our body also signals our brain to get warm by putting on a jacket. The opposite happens if we get too hot. Our body tries to cool down and sweat is released to aid in that process. The water pulls the heat away from our skin, causing our body to cool down. Then, of course, we probably get thirsty so that we can replenish the water that we have lost. But homeostasis is not just about temperature. The amount of nutrients, salt, and water balance is also very important in maintaining homeostasis. I remember in college, I had a professor who was very healthy and kept a very low sodium diet. But one day we were waiting for her in class and she didn't show up. So we wondered what had happened to her. We found out later that she was biking to class and just collapsed off her bike and was sent to the emergency room. So what happened? For most of us, a low sodium diet is a good idea because we get too much sodium in our food. But for her, she was overcompensating with her strict diet and went too low in her sodium level. So losing consciousness was a way that her body was warning her that she needed to eat more salt. Living organisms have a very narrow range and must maintain internal constancy in order to stay alive. The next characteristics of life is reproduction, growth, and development. There are two kinds of reproduction: asexual or sexual reproduction. In asexual reproduction, the offspring only has one parent, and this offspring is genetically identical to that parent. It's basically a clone of the parent. If you look at this strawberry plant, it sometimes reproduces asexually. That means if you looked at some of the offspring of that plant, you'll find that they're identical to the one parent plant that it came from. The DNA would be pretty much exactly the same. This is an example of asexual reproduction. Asexual reproduction is mostly beneficial if the organisms are in an environment that is constant and unchanging. For example, if the temperature, precipitation, predators, etc. tend to always be the same. Then organisms that undergo asexual reproduction can flourish and successfully reproduce. The second type of reproduction is called sexual reproduction. Offspring from this type of reproduction have two parents, and they are not identical to their parents. These offspring get half of the DNA from each parent. Plants and animals reproduce through sexual reproduction. And the baby swans or cygnets that you see in this picture have also reproduced sexually from their parents. In this case, these cygnets are genetically different from their parents, which means they have features that are different from their parents, and they also have features that are different from one another. That's why siblings are similar, but not exactly the same. Unlike asexual reproduction, sexual reproduction is more beneficial for a population when the environment is constantly changing. Since there's more diversity in the population, this increases the probability that at least some organisms in that population may survive, especially if something happens in the environment that causes many organisms in that population to die. For example, droughts, hot temperatures, too much rain, a new predator, or sudden change in the surroundings could all contribute to some organisms not being able to survive. Now, even though there are two types of reproduction, asexual and sexual, both types of offspring shown here began as one cell and grew into multicellular organisms that are composed of many cells. For example, all of us started off as one cell that was formed when a sperm and an egg came together. Okay, I think we're ready for another question. Go ahead and pause the video to answer the following question. So we just went over the four characteristics of life: organization, energy, internal constancy, and reproduction, growth, and development. The last characteristic of life is that life evolves. Evolution is defined as genetic change over time in a population. Notice the bacteria shown here. The red color represents a bacterial cell that has undergone a mutation or a change in its DNA composition. This random mutation has given this red bacteria a new function, the ability to confer antibiotic resistance. What about this pygmy seahorse? Notice how the seahorse blends in perfectly to its environment. How did it get that way? Is it that the seahorse decided that it would survive the best if it changed its color to fit the coral habitat? That would be nice, but that's not actually how evolution works. There's actually a certain version of genes in the seahorse that gives it this color combination. So how did the seahorse get this way? Imagine that there were many different colored seahorses in this habitat at one point. There are black ones, white ones, blue ones, green ones, etc. Now, one day some predators come along because they're hungry, and they wreak havoc by trying to capture all the seahorses. Which ones do you think gets eaten first? Yes, it's probably the ones that the predators can see best. So over multiple generations, the seahorses with colors that stand out generally get eaten, and the ones that blend in better with the background tend to survive. After multiple generations, you find that the seahorses that have survived and successfully reproduced are the ones that can blend into that environment. This is an example of evolution by natural selection. Let's go back to the bacteria again. As you may already know, bacteria reproduce very quickly, every few minutes for some species of bacteria. So you can imagine that they have millions of offspring in a short period of time. As they reproduce over many generations, there's a higher likelihood that by random chance, mutations or errors that occur in DNA can accumulate and give rise to some change in the bacteria. In this case, the mutation gives rise to antibiotic resistance. The red bacteria has randomly become resistant to antibiotics, again, just by chance. Now, remember this bacteria is reproducing asexually. So as long as this bacteria is in an unchanging environment, it'll be very successful at reproducing and surviving. That's shown here in the bottom right after multiple generations where antibiotics are absent. However, what happens when all of a sudden this bacterium is in an environment with antibiotics? That would happen if you got sick with a bacterial infection, went to the doctor, and got a prescription for antibiotics. In your body, the bacteria are now in trouble, and almost all of them will die. This is good for you. But once in a while, there could be one bacterium that develops resistance to that antibiotic, again just by chance. And this one bacterium will survive. Now, this is okay if reproduction is slow, but remember some bacteria can reproduce every few minutes? That means even though the green bacteria has mostly died off from the antibiotics, soon the antibiotic-resistant bacteria, shown in red here, will reproduce, infect the cells in your body, and get you sick again. Now, these bacteria are resistant to the antibiotic that you took originally, so if you continue to stay sick, your doctor might have to prescribe another antibiotic for you to try, hoping that there are no bacteria left that are resistant to the second antibiotic. So then we have covered the 5 characteristics that biologists use to determine life: organization, energy, internal constancy, reproduction, growth and development, and evolution. At this point, you should be making connections between the terms that have been introduced here. For practice, see if you can make a concept map that connects all these terms together and form relationships between them. Now that we have defined the characteristics of life, we must also look at how life is categorized. There's so many different kinds of living species, so we have to have a good strategy on how to classify them so that we can study them in an organized way. Taxonomy is the term we use for naming and classifying organisms. All living organisms fall into one of 3 categories within what we call a domain. There are 3 domains: bacteria, archaea, and eukarya. All 3 of these domains then have one or more subdivisions called kingdoms. The eukarya domain has the most kingdoms and probably ones that you recognize, such as animals and plants. The domains bacteria and archaea are what we call prokaryotic, and most of the organisms in these 2 domains are made of one single cell, or are unicellular. Organisms in the bacteria and archaea domain are also prokaryotic, which means that these organisms do not have a membrane-bound nucleus that surrounds the DNA. These species have DNA that are just in the cytoplasm of the cell without a nucleus. Eukaryotes, on the other hand, are organisms that have cells with DNA inside a nucleus, so the DNA is membrane-bound. That's the main way that we can differentiate between organisms that are prokaryotic versus eukaryotic. Notice here again that most of the organisms that are in the bacteria and archaea domains are unicellular, and all of the organisms in these two domains are prokaryotes. On the other hand, organisms in the domain eukarya have cells that have nuclei. This is just plural for nucleus. In the kingdom Protista, you have organisms that can either be single-celled or multicellular, meaning that the organism is made up of many cells. This includes organisms like amoebas and algae. Organisms in the domain Protista can either be autotrophs or heterotrophs, which are categories based on whether or not the organism produces its own food or feeds on other organisms for food. Plants, protists, and some bacteria are autotrophs and make their own food source through processes like photosynthesis. Heterotrophs, in contrast, feed off other species to get their food. There are protists that are heterotrophs and protists that are autotrophs. In the domain Eukarya, there's also the kingdom Animalia. That's one that you're probably familiar with and includes vertebrates like fish and frogs, and invertebrates like bees. These organisms are multicellular and heterotrophs by ingestion, which means they eat their food. These organisms are also composed of eukaryotic cells, which all have a membrane-bound nucleus. The next one is the kingdom Fungi, or sometimes pronounced fungi. Actually, I have a joke that relates to this kingdom. A girl mushroom and a boy mushroom went out on a date, and after the date was over, they were at the door saying goodbye. What did the girl mushroom say to the boy mushroom? You're a fungi. Okay, so in this kingdom, most of the organisms are multicellular. Although there are a few organisms like yeast that are unicellular, fungi are also heterotrophs, but by external digestion instead of digesting internally. They absorb nutrients from the environment and are nature's decomposers. Lastly, there's the kingdom Plantae, where plants are categorized. These species are all multicellular and autotrophs, again meaning that they produce their own food source. In this case, they produce food through a process called photosynthesis. Okay, let's see if we can answer this question about the difference between eukaryotes and prokaryotes. Feel free to pause the video. Again, you should be able to make connections and know the relationships between the terms that are listed here. See if you can come up with a concept map that highlights the relationships between these words. Now that we have defined life and categorized it, we're going to talk about how we go about studying living organisms. Scientists follow a systematic approach that we call the scientific method. Each step is related to one another. Let me take you through the steps starting with observation. Let's say you made an observation of something in the natural world. It could be anything that you observe with one or more of your five senses. You may notice that the sky is blue, the trees have green leaves, that flowers are blooming in the spring, and so forth. So then this observation may lead you to a question. Let's use a practical example that may relate more closely with your life experience. Say you were on your way to school and noticed that when you put your key in the ignition of your car, it doesn't start. What kinds of questions might you ask yourself? What's wrong with my car? Why isn't it starting? Did I put in gas last night? Is there something wrong with the starter? Why is the car making this funny sound? So let's pick one of the questions as a research question. How about, why isn't my car starting? At this point, you'll probably be consulting prior knowledge by looking at the car manual, calling your mechanic friend, thinking about what your brother did when his car didn't start, and so on. This will eventually lead you to come up with some probable solutions to your problem. The research question will lead you to formulate a hypothesis. A hypothesis is a tentative explanation for observations, a possible answer to a question that you're asking. It also needs to be a testable hypothesis, since it's a possible answer to the question. The hypothesis is in the form of a statement. How might we answer the question, what's wrong with my car? One hypothesis might be, my car battery is dead. This will lead us to make predictions that will allow us to collect data or conduct an experiment. Predictions are usually in the form of an if-then statement and leads us to an experiment. Here's an example. If I change the battery on my car, then the car should start. This prediction leads me to try this out in an experiment. So I change the battery and see what happens. The car starts. What's my conclusion then? In a scientific experiment, the result of an experiment does one of two things. Either supports or falsifies the hypothesis. However, since our car is now starting, can we conclude that the problem was definitely the battery? What if we put the old battery back in and the car starts again? Then we might be led to think that the problem was not the battery after all. Could it have been that there was too much crud on the connections to the battery, and when we changed the battery, we cleaned this off and made the connections better? At this point, we may never know for sure what exactly the problem was. We just know that it works now, and we support the hypothesis that the car didn't start because the battery is dead. But we still don't know for sure if that hypothesis is definitely true. In fact, this is how scientific investigations work. We never prove that the hypothesis is absolutely true without a shadow of a doubt. We just support or falsify the hypothesis, and as we and others do many experiments, if the hypothesis continues to be supported, it may move up in level to a scientific theory or even eventually to a scientific law. However, even laws can be rejected or falsified if there's substantial evidence from experiments that reject the original hypothesis. One example is that scientists believed for a long time that the world was flat, until we had the technological advances that gave us more evidence that the earth is round. As we continue to advance in our knowledge of the natural world, what we know about the world will continue to change. So we can support or reject hypotheses using the scientific method and continue refining what we know about the natural world. But remember, in science, we don't ever say that something is definitely proven to be true forever. We leave it open for more scientific evidence to confirm or refute it. Once a scientific experiment is complete, if there's enough data to convincingly support or falsify a hypothesis, scientists can submit a manuscript to a peer-reviewed journal. Then the editors of the journals and other experts in the field will decide if the research finding is worthy of being published in their journal. In our example of the car, we probably would not decide to publish. When scientific experiments are being conducted, there are two different ways of making scientific investigations. One is called discovery science, and the other is controlled experiments. In discovery science, you're mostly using your observation skills and collecting data on what you see in the natural world. You're not manipulating anything. For example, you might be interested in migratory patterns of birds, which might lead you to take pictures or count the number of birds that are in a given region at a certain time of the year. Then you would analyze this data. You could collect data that showed how many birds are in this area from January to March versus April to June. In controlled experiments, you have variables such as independent, dependent, and standardized variables. In this picture shown, the investigators could be trying to figure out which type of coffee bean has the most caffeine. In this case, the independent variable would be the different kinds of coffee beans you're using, because you as the scientist are selecting or manipulating this variable. You're using different kinds of coffee beans. In other cases, you might decide to look at differences in sizes of coffee beans, the fragrance, the color, and other variables that you could use as the independent variable. In this case, the independent variable is the type of coffee bean. Now, the dependent variable would be what is being measured, or the data that's being collected from the experiment. In this case, they are looking at the amount of caffeine in the different kinds of coffee beans. So the dependent variable would be the amount of caffeine. Lastly, all the standardized variables are variables that we keep the same throughout the experiment. In this case, we'd make sure that the mass of the beans, the cup used, the machines used to brew the coffee, the tools used to measure the caffeine, the expiration date of the coffee beans were all the same. We want to keep as many variables standardized as possible so that we know that the result we find is due to the independent variables. A well-designed experiment also includes controls and test groups. The control group is the group that's being used for comparison. You can have negative and positive control groups as well. The negative control is the group that should give you a negative result. The positive control is the group that should give you a positive result. You can use these controls to compare with the experimental groups. The experimental groups are the ones that receive the treatment. These groups may or may not show results that are different from the control groups. In this next question, let's see if we understand the difference between the independent, dependent, and standardized variables. Here's data from a study about the effectiveness of rotavirus vaccinations for infants. We see that there are variations in the amount of vaccine that was given to the infants. None are placebo, low, medium, or high dosage of the vaccine. This graph gives us incidence of illness, or how many babies got sick. Which letter represents the dependent variable in this experiment? See if you can explain why that's the dependent variable. Let's try this next question as well. Feel free to pause the video to answer the question. At this point, you should feel somewhat comfortable with the scientific method and how it works. Remember, I mentioned that all hypotheses are refutable if there's experimental evidence that can falsify the hypothesis. However, if hundreds of experiments have been done for many years on a certain hypothesis, then we can begin to elevate this hypothesis to what we call a scientific theory, or a broad explanation for a natural phenomenon. It can eventually be even more elevated to a scientific law, like the law of gravitation. Now you should try to see if you can differentiate between the terms hypothesis, scientific theory, and a fact. A fact is a statement that's based on observations of the natural world that's usually indisputable, such as the sky is blue. Think about how non-scientists use these words and how that might be confusing if they're not explaining it clearly. Here's a real-life example of how scientific theories help explain observations. In an observation that was made by Charles Darwin, he saw nectar tubes on orchids that were really deep and narrow. Based on his theory of evolution by natural selection, Darwin predicted that there must be an insect that had an equally long tongue that can reach inside the nectar tubes. In order for these plants to survive, reproduction and fertilization had to occur through pollination, often from insects. This meant that the insect that pollinated this orchid must be able to reach the nectar, since that's probably what attracted the insect to the orchid. If the insect was unable to reach the nectar, it may not have come back to visit the orchid, and the orchid would not have survived and reproduced. So Darwin predicted that there had to be an insect that had a long tongue that can reach the nectar, even though he had never seen one before. This prediction was based on his theory of evolution by natural selection. These insects with longer tongues were more likely to survive because they can reach the nectar in these orchids and had exclusive access to food that other insects could not get. So the orchid and the insect continued to evolve together until only these insects were able to pollinate this type of orchids. It wasn't until 30 years later that a moth was actually discovered, and indeed, its tongue was super long and able to reach the nectar inside the orchids. As you reflect upon the scientific method, think about what kinds of observations Darwin and Wallace must have made in order to make their predictions about the existence of a long-tongued moth in Madagascar. Even though the scientific method has the potential to give us many explanations for scientific phenomenon, it also has some limitations. First of all, the data from the experiments could be interpreted in more than one way. When we talked about the car battery example, there were multiple possibilities for why the car had not started. We supported the hypothesis, but were not able to rule out other possibilities that also may explain what had happened, such as loose connections, the starter, and so forth. Another limitation of scientific inquiry is that the data can be viewed skeptically for non-scientific reasons. For example, if we were making conclusions about the natural world while looking through a religious lens, we would come to different conclusions than what would be found using the scientific method. Lastly, some hypotheses just can't be tested. Hypotheses that are based on morality or opinions fall into this category. Here's an example. Just because the moth visited the orchid often, it doesn't mean that we can come to the conclusions that the moth likes the orchids. We can't ask the moths if they like the orchids, and just because they visit often doesn't necessarily mean that they like it. For example, I'm sure many of you come to school often, but there's a chance that you may not like school very much. So even though the scientific method gives us explanations for natural phenomena, we need to be aware of some of the limitations that we have in scientific inquiry.